Hello, welcome to our discussion of bioenergetics. Remember when we first started the course, I talked to you guys about metabolism. And when I say metabolism, I want you to think about the processes or chemical reactions called photosynthesis and cellular respiration. And bioenergetics is the study of how those reactions occur. This beautifully relaxing vision is of the plains of the Great Midwest, or the Great Plains of the Midwest in the United States. And of course, I grew up out in Oklahoma, and if you weren't looking at cornfields, you were looking at big open wheat fields, just like this. Big sky. Anyway, what distinguishes us from the inorganic realm is the ability to cycle matter and energy. And so where do we get energy? Of course, this course is an herbivore, but I think herbivores like horses and cattle will sometimes feed on dead animals just to sort of obtain excess protein. So yeah, the horse is an herbivore, but don't try to pet the wild horse. We use chemical energy to sustain our life processes. And there's a couple kind of changes that happen with matter. One is a physical change. You can see here to the right, I got a block of ice that's being heated up. And of course, I can convert that solid water right to gaseous water in a process called sublimation. Just underneath, I see an example of the BZ reaction. And it's an example of a chemical change. And in this chemical change, the atoms literally are getting rearranged. The BZ reaction is interesting just because it shows this ability for the inanimate to self-organize. Not super organized, but kind of organized. Of course, phase changes, physical changes, moving from gas to water. There's no rearrangement of atoms. It's just the shape of the stuff has changed. And in chemical changes, we rearrange atoms. So that is physical versus chemical changes. And of course, if we're going to talk about bioenergetics, we're going to refer back again to this science called thermodynamics. And thermodynamics is, again, the study of how energy and matter interact and change. And the first two laws, there's actually 4 laws of thermodynamics. So the first 2 laws are important for our discussion here of enzyme activity. First, law, thermodynamics, delta U equals Q minus W. It's the total heat of the system is equal to the heat of the system minus the work that's done. In short, it says that matter and energy isn't created or destroyed, and in fact, every time there's a transformation, energy is lost to the universe's heat. And this blends right into the second law of thermodynamics. The second law of thermodynamics relates to the concept called entropy. And the second law says that the entropy, or S of the universe, always increases. Entropy is simply a measure of the randomness or disorder of matter and energy in a system. Just how disorderly the system is its S value. And we'll think about both of these topics when we talk about enzyme reactions. So the question is, how does this GIF relate to the first law? And I'll leave that one to you. When we talk about chemical reactions and chemical changes, we have to talk about something called the energy of activation. Okay, so in general, when we think about chemical reactions, you think about reactants, some reactant A plus B that join together form AB or C, and we call these reactants and products, and we form bonds between those atoms, and they could be covalent or ionic, but that's not what we're here to talk about. This GIF shows us a wonderful explosion that happened right in Philadelphia this past summer, 2019, and it's an example of what kind of a chemical reaction. So when we talk about chemical change or chemical reactions, there's a concept called the energy of activation. And the energy of activation is the minimum energy needed to start a chemical reaction. So there's two factors that limit the ability for a reaction to occur. Number one, the reactants have to get together, come together with enough energy for them to form a bond. And remember that atoms have a shell of electrons that is negatively charged. So everybody's got electrons, more or less. And when they come together, there's got to be enough energy for those two atoms to collide to overcome that natural repulsion between the electron shells. Not only that, but they also have to be in the appropriate orientation. Now, that's a little bit deeper than we need to go for the course, but those are the two factors that limit chemical reactions. When we diagram chemical reactions, one of the ways we'll use to help us understand changes in energy is through something called Gibbs free energy. Gibbs free energy is a way for us to talk about the amount of energy that's stored in chemical bonds and whether or not that energy or the amount of Gibbs free energy in a system between the atoms has increased or decreased. Delta in the sciences and maths always equals change. And so if I have a negative delta G, I've gone from an area where there's lots of energy stored to a place where there's not so much energy stored. Of course, on the other hand, if I go with a positive delta G, I've stored energy. And we can describe reactions in terms of their delta G as being either endothermic, also called androgonic, or exothermic, or exergonic. Now, exothermic makes sense to me. I'm giving off heat. Exothermic, give off heat. Endothermic, of course, means inside heat. So hopefully those two terms will help you remember what kind of reactions are which. Down bottom, we have some graphs, and I want you to tell me, when we talk about metabolism, I want you to think of photosynthesis and cell respiration. Which of these graphs belongs to cell respiration? Which one belongs to photosynthesis? Which one's endothermic? Which one's exothermic? Hopefully you can figure that one out. Now, metabolism can also be spoken about in terms of, in fact, metabolism explicitly means the sum of all chemical reactions, and chemical reactions can really fall into two categories, anabolic or catabolic. Now, anabolic reactions generally take smaller molecules and add them together. Ana means to add, all right? So I make big molecules out of smaller molecules. In catabolic reactions, I break down larger molecules to smaller molecules. Generally speaking, anabolism will give off energy. To build things up takes energy, and when I break stuff down, I will release energy. And here's some examples from our biochem unit. So remember, when you think about anabolism, you think about building larger molecules using energy. And the perfect example of that are dehydration synthesis reactions. Remember, we made polymers through dehydration synthesis. Dehydration synthesis produces a water molecule. Every time we join two monomers together, we get a little water molecule. And this is, of course, is how I make polysaccharides. Proteins, right? Chains of amino acids. The triacylglycerides. This is how I make chains of nucleic acids, or nucleotides or nucleic acids. On the other hand, an example of catabolism is when I break down big molecules into smaller ones, and the classic reaction is hydrolysis. Remember that I can take a chain of starch and cleave off individual glucose units by actually adding water to the reaction. So hydrolysis, I break apart by adding water. Dehydration synthesis, I build things by producing water. Down the bottom here, you can see a guy who's probably using anabolic steroids, and then another guy who's not, but I don't know which one you like better. Remember that biological reactions involve very large molecules, and these large molecules have require large amounts of energy in order for them to be built up or broken apart. And so without enzymes, these reactions just simply wouldn't happen quickly enough. In any individual cell, you might have 10 million reactions happening per second. 10 million reactions per second. And remember, you're made up of 70 trillion cells, so it's virtually an infinite number of chemical reactions. And we can't just rely on luck. Remember that, generally speaking, in a chemical reaction, we have some reactant A and some reactant B. They have to hit each other with enough energy and in the right position. It's all luck. It's a random thing. I can't rely on luck to allow those 10 million reactions to move ahead. And so the answer for living systems are little proteins called enzymes. Enzymes, remember, are proteins. They end in ASE. And enzymes are biological catalysts. Catalysts are substances that can speed up chemical reactions by lowering the energy of activation. So there's the test question, right? Enzymes increase the rate of reactions by A, increasing the energy of activation, or B, decreasing the energy of activation. Now, there's a graph up top here. It's that energy on the y-axis refers to Gibbs free energy. And then on the bottom, we simply have time. And there are two reactions, one in red, one in blue. The one in red is the reaction without an enzyme. The one in blue is the reaction with an enzyme. You'll see that the energy of activation has been reduced by the enzyme. How does it actually do it? Well, enzymes are proteins. They are large molecules that fold up in a very specific shape. And on the enzyme, there is a place called the active site. Active sites bind substrates. And one of the interesting animations on this slide is down bottom. I want to bring your attention to that. Notice two H2O2s. That means two peroxides. And it produces two waters and an oxygen molecule. You guys have probably seen this reaction before. What enzyme is involved? So remember, biological reactions proceed due to the activity of enzymes. Enzymes catalyze the reaction by lowering the rate of, the activation energy. And enzymes have little special places. They're large proteins and they have little special places called active sites. Enzymes bind their substrates. That's their target at the active site. And then they change their shape a little bit and they actually will catalyze a reaction, whether it's putting something together or tearing things apart. And that change in shape of the enzyme, once it binds its substrate, is called allosterism, and it's related to something called the induced fit model. Enzymes are also very sensitive to changes in temperature and pH. Generally speaking, most reactions will speed up as I increase temperature. But with enzymes present, there's a limit to that. And of course, pH is important in biological systems. And if I change pH, I'm changing hydrogen ion concentrations. Remember, what holds a protein together? Well, we could go deeper and deeper into the topic, but generally speaking, we allow biological reactions to proceed by using enzymes. That makes the reaction speed up, sometimes by a millionfold just by the addition of an enzyme. And remember, every time a chemical reaction occurs, there's energy lost to the universe's heat. Let's see, by obeying that second law of thermodynamics, or the first law. Well, what biological systems consistently do is they trap that extra energy in the form of a molecule called ATP, and it is the energy money of the cell. And of course, I'm going to ask you to give me an example of how ATP is used to do some useful work. We're going to learn a lot about ATP soon.